This week, we'll learn how to engineer DNA so that it instructs cells to make a target biologic that we can harvest and use as a drug. The protein we want is usually not produced by the host natively, so this process is called heterologous protein expression. Creating a suitable expression system is typically referred to as cell line development and is part of the so-called upstream process development. There are several steps involved in cell line development. In this video, we'll look at the steps broadly, and then in later videos consider each step in greater depth. The first step in generating a cell line for protein expression is to choose both the host cell line and an expression vector. An expression vector is just an engineered DNA construct used to put a foreign gene, such as our target biologic, into the cell. Choosing a host is extremely important because it'll affect almost every aspect of production. Whether we end up with a quality product, how that product is produced and purified, how quickly a production process can be developed, and of course, how much our manufacturing process will cost. After we've chosen a host, we have to get the expression vector that codes for our target molecule into the host cell. This process is called transfection for eukaryotic cells and transformation for bacterial cells. Ideally, for eukaryotic cells, we'd like our host cell to incorporate the vector into its genomic DNA, rather than just having it floating around inside of the cell. This stable integration allows continued expression of the target biologic over millions of cell divisions and is necessary for commercial manufacturing. Transfection can also be transient or temporary. In transient transfection, the vector is not integrated into the host DNA, and the gene of interest only replicates over a few rounds of host cell divisions. So the target biologic will only be expressed during a limited number of generations of the host cells. Transient transfection, although unsuitable for commercial manufacturing, is useful in the early stages of process development when the manufacturing process itself is still being worked out and the clinical utility of the biologic is still unknown. Unfortunately, the transfection process is not efficient. In other words, most cells do not stably integrate the vector into their DNA. So when we design a manufacturing process for commercial use, we need to somehow select only the host cells that have integrated the vector and kill the rest. There are many ways to do this. For example, one is to add a gene to the vector that allows the host cell to resist a toxin. The gene may code for an enzyme that neutralizes the toxin, allowing the host cells to survive. Adding that toxin to the growth media will select for cells that integrated the vector with the gene for our target. The other cells will die. This selection process ensures that all the surviving cells have taken up the vector, but some might have more copies than others, or may have modified the biologic by deleting it or truncating it. So we need to choose one cell, a single clone that produces our target biologic most effectively and with the best quality. Once we've chosen that one cell, we'll allow it to reproduce and we'll have our master cell line. Typically, we'll evaluate a few candidate clones for their ability to stably produce the biologic at suitable concentrations, or titers, under production conditions similar to those that we'll use in the final process. We'll pick the best clone and allow it to divide until we have billions of cells. Just like you might make a large batch of dough for bread and freeze portions of it for later use. We'll split up the cells and freeze hundreds of individual batches. This master cell bank must provide the cells that will manufacture the biologic over its entire commercial lifetime. In the remaining segments for this week, we'll look at each of these steps in more detail, review some of the history related to the types of cells commonly used in manufacturing today, and consider some of the emerging advances in technologies in cell lines.